Hello and welcome to my channel Sam Biology Class. Friends, today we will discuss about shotgun sequencing. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. In biology, sequencing is a process. In biology, sequencing is a process that is used to determine the order of nucleotides in DNA and RNA. This technique is critical for understanding genetics, for identifying diseases, and also for tracing ancestry. There are different methods of DNA sequencing. One of them is Sanger sequencing. One of them is Sanger sequencing. This technique was developed by Frederick Sanger. This technique was developed by Frederick Sanger in 1977. This technique is also known as chain termination sequencing, is also known as chain termination sequencing, or dideoxy sequencing, or dideoxy sequencing. This name comes from the use of modified dideoxynucleotides that terminates DNA synthesis when incorporated into the DNA chain. Sanger sequencing is useful for sequencing small DNA fragments. Sanger's method is best for sequencing DNA fragments up to approximately 800 to 1000 base pairs. Larger fragments cannot be sequenced by this method at a time. This technique can determine the sequence of nucleotides in one fragment at a time. So this process is slow and expensive for large-scale sequencing projects. Sanger's method is not suitable for sequencing genomes. This is because of its complexity and cost. In addition, it is also time-consuming. The sensitivity of Sanger sequencing is low. In addition, this process also requires high quality and purified DNA sample. Now the question is how to sequence genome. The answer is shotgun sequencing. The answer is shotgun sequencing. Shotgun sequencing is a method that is used to sequence long DNA molecules, chromosomes, and even entire genomes. This technique involves random fragmentation of genomic DNA into many small overlapping fragments. These fragments are then sequenced by Sanger sequencing or next generation sequencing. In next step, the sequenced fragments are reassembled based on their overlapping region by using computer algorithms to reconstruct the original sequence of the genome. The concept of shotgun sequencing was proposed by Roger Staden. In 1979 to speed up the process of genome sequencing. He also developed the first DNA sequencing software. In 1981, John Messing, he developed the first shotgun sequencing protocol using M13 phase vector. In 1982, Frederick Sanger, who developed the chain termination method, used shotgun sequencing to sequence the genome of Bacteriophage lambda. In 1995, J. Craig Venter and Hamilton Smith used whole genome shotgun sequencing to sequence the genome of Haemophilus influenzae. In late 1990s, Venter's private company called Celera Genomics used shotgun sequencing approach to sequence human genome and collaborated with Human Genome Project. The name shotgun comes from the random explosive-like fragmentation process similar to a shotgun blast. A book example similar to shotgun sequencing is shredding the book at random into a large number of strips. The strips are then reassembled using overlapping words and phrases to reconstruct the entire book. In this example, the book represents the genome, and shredding is the first fragmentation step. The librarian who pieces together all the strips based on the overlapping act like the computer program. Shotgun sequencing is a highly efficient process that is widely used to sequence long DNA molecules, chromosomes, and entire genomes. Now let us discuss about the different steps of shotgun sequencing. First step is the extraction and purification of genomic DNA. In second step, the genomic DNA is broken down at random to produce a large number of overlapping DNA fragments. So second step is random fragmentation. Fragmentation of genomic DNA can be achieved by either mechanical shearing or enzymatic digestion digestion. Mechanical shearing can be done by different processes such as sonication, nebulization, and focus acoustic shearing. In addition to mechanical shearing, the DNA can also be fragmented by using specific enzymes such as restriction endonuclease, or DNase1. Fragmentation is followed by DNA sequencing. All these fragments can be sequenced by either Sanger's method or by next generation sequencing NGS. The main difference between these two is throughput. While Sanger's method can sequence only one DNA fragment at a time, NGS can sequence millions of DNA fragments simultaneously. In addition, NGS has a sensitivity that is higher than the Sanger's method. As NGS is much faster and efficient than Sanger's method, it has replaced the role of Sanger's method in shotgun sequencing. Sequencing is followed by sequence analysis and reconstruction. As we have a very large number of overlapping DNA fragments, manually it is not possible to reassemble them in order to reconstruct the complete sequence. For this, we take the help of computer program. All the sequence fragments are analyzed, and then based on their overlapping regions, the complete sequence is reconstructed. For example, we have 3 DNA fragments, and these are the overlapping regions between them. Based on these overlapping regions, we can place 3 different DNA fragments one after another in this particular sequence. In shotgun sequencing, computer program analyzes the sequences of a large number of DNA fragments known as reads and find out overlapping regions between them. Then by aligning the overlapping regions, the complete sequence of the genome is constructed. By using computer algorithms, various sequenced DNA fragments are reassembled based on the overlapping regions into larger sections called as contigs. These contigs are then assembled in a specific manner to form the complete chromosome, or scaffold. So friends, in this way, by using specific computer programming, shotgun sequencing can determine the arrangement of nucleotides in complex genome. Applications of shotgun sequencing. Number 1. Shotgun sequencing is widely used in biomedical research, evolutionary genomics, and in analyzing complex microbial communities. Number 2. This technique is also highly effective in analyzing highly degraded and low quality DNA sample which may be present in ancient genomic and also in forensic traces. Friends, this is all about the principle and applications of shotgun sequencing. For more updates, subscribe my YouTube channel, Sam Biology Class.